COVID-19, influenza, and other global pandemics have demonstrated the great power of pathogens in affecting human health. But pathogens can also threaten humanity in other ways. Infectious diseases in plants, such as the rust pathogens that infect wheat, barley, and other grains, can reduce crop yields. Others, such as the Fusarium wilt, attacking bananas, threaten to wipe out entire cultivars. And unlike human diseases, few to no medicines cure plant diseases. But just like humans, plants have a natural immune system.
Plants have an amazing ability to recognize pathogens and fight back.
Fascinated by the immune system of plants, molecular geneticist Jian-Min began studying the proteins involved in plant immune responses while he was at the National Institute of Biological Sciences in Beijing, NIBS, in the early 2000s. Scientists knew that inside plant cells, a group of proteins called NLR receptors played a role, but did not know how they trigger immune responses. To solve this mystery, Jijie, a structural biologist who joined NIBS in 2004, began a research partnership with Jian-Min.
NLR receptors are pivotal in facilitating plant defense against pathogen infection.
After moving to the genetics and developmental biology of the Chinese Academy of Sciences, Zhou's team identified the function and mechanism of an NLR receptor called ZAR1 in mustard plants and found that it worked with two other proteins, RKS1 and PBL2, to detect signs of infection from bacteria that cause black rot.
Amazingly, this set of proteins turned out to be an ideal model for reconstitution and structural study of plant NLR-containing complexes by Professor Cai's group.
While at Tsinghua University, Zhai's team used a technique called cryo-electron microscopy to visualize ZAR1's molecular structures. They found that, when inactive, the ZAR1 receptor holds a molecule of adenosine diphosphate, ADP, in its center. When bacterial proteins are present, they react with and enable PBL2 to bind to the RKS1 protein at one end of ZAR1. This forces ZAR1 to exchange its ADP molecule for deoxyadenosine triphosphate, DATP. ZAR1 then becomes active and assembles in groups of 5, forming a star-shaped complex, which the researchers term a resistosome, referring to a group of plant proteins conferring resistance to a plant disease. The structure of the ZAR1 resistosome gave the researchers clues to its function.
This was the first time that anyone had ever seen a resistosome. We observed that the resistosome exhibits structural features resembling a channel or a pore.
Cell membranes are often riddled with channels to allow various compounds or ions to pass through. Zhou's team tested and confirmed their hypothesis that the ZAR1 resistosome could interact with cell membranes to form a pore.
It was indeed an ion channel that allows calcium ions to enter plant cells. In turn, the increased concentration of calcium ions sets off plant immune responses.
Armed with a new understanding of the plant's natural immune system, the Zhai team, now at Westlake University, and the Zhou team have begun to engineer NLR receptors to gain further insights into how plants defend themselves. As the world's population grows and climate change and global travel are expected to worsen plant pathogen outbreaks, understanding and improving the immune systems of our crops cannot come soon enough.
By helping plants recognize a wide range of pathogens, we hope to substantially enhance the ability of crops to combat diseases.
For the discovery of plant resistosomes and their roles in plant immune responses against pathogens, the 2023 Future Science Prize in Life Science goes to Jijie and Jian-Min.